Today's video presentation will cover information pertaining to tubular reabsorption, particularly the reabsorption of water and urea. Per usual, the first few slides will discuss general information about the different mechanisms of reabsorption, as well as examples for some of those mechanisms. The rest of the presentation will then focus on the main concept, which will be the reabsorption of urea and water. There are several different mechanisms that involve the movement of substances across membranes, either for reabsorption or secretion. Those mechanisms include diffusion, facilitated diffusion, active transport, secondary active transport, and osmosis. Diffusion is simply a process by which substances move from an area of higher concentration to an area of lower concentration. Because the substances are moving with the concentration gradient, this mechanism does not require any energy. Facilitated diffusion also moves substances down the concentration gradient. The difference, though, is that this certain mechanism requires general proteins or membrane receptors for movement. Glucose and sometimes sodium ions are moved across the membrane through facilitated diffusion. There are certain cases of mediated transport where the same general protein port is used by two different protein substances. Those mechanisms are referred to as symport and antiport. In contrast to diffusion, the mechanism that moves substances from an area of low concentration to high concentration is called active transport. Because substances are moved against the concentration gradient, this mechanism requires energy from adenosine triphosphate, or ATP. A good example of active transport is the sodium-potassium pump, which transports sodium out of the cell and potassium into the cell. Secondary active transport is a specific form of active transport. This mechanism moves ions, particularly sodium and hydrogen ions, coupled to transporter proteins. The last mechanism I have mentioned that transports substances is osmosis. Osmosis is just the simple diffusion of water. It is important to know and comprehend the previously discussed mechanisms because it helps provide a better understanding of the processes of reabsorbing water and urea into the bloodstream. The reabsorption of water and urea occurs passively following the movement of osmotically active solutes. This is to ensure that the end of the proximal tubule contains a fluid that is isoosmotic with plasma. What is tubular reabsorption? Tubular reabsorption refers to the process of removing water and solute from the tubular fluid or pre-urine and returning them to the bloodstream. The process is termed reabsorption and not absorption because the substances involved have already been absorbed once in the digestive tract. Also, the substances are being reclaimed from a post-glomerular fluid stream that is soon to become urine. The return of reabsorbed water and substances to the bloodstream is achieved by the peritubular and vasa recta capillaries. As before mentioned, the reabsorption of water occurs passively through the tubule. Two of the factors that affect the reabsorption of water into the vasa recta and peritubular capillaries are concentration gradients and osmolarity. Sodium actively transports out of the proximal convoluted tubule, or PCT, to be able to enter the interstitial spaces between the cells. Sodium then diffuses down the concentration gradient straight into the peritubular capillaries As sodium diffuses, water follows passively to try and maintain an isotonic fluid environment within a capillary. The process by which water osmotically follows actively reabsorbed sodium is known as obligatory water reabsorption, simply because water is obliged to follow. Approximately 65% of filtered water is passively reabsorbed in the PCT. That is equivalent to about 117 liters of water each day. Another 15% of filtered water is passively reabsorbed, but instead of going to the proximal tubule, it passively reabsorbs in the loop of Henle. This 80% of passively reabsorbed water is obligatorily reabsorbed no matter the water load in the human body. This means that there is still 20% of filtered water unaccounted for. This 20% is reabsorbed in the distal tubule. The extent at which water is reabsorbed in the distal and collecting portions of the tubule is mainly controlled by hormones. Certain parts of the nephron, like the loop of Henle, become less permeable to water and solutes. However, with antidiuretic hormone, water permeability greatly increases in the distal and collecting tubules. Another factor that contributes to this extent is the body state of hydration. Understanding some of the general information about water and percentages of how it can be reabsorbed, let us now talk about what actually occurs during the process of reabsorption. The first step of water reabsorption involves the passing of water through water channels, otherwise known as aquaporins. Those water channels are formed in the tubular cells by specific plasma membrane proteins. There are different types of aquaporins in various parts of the nephron. In the proximal tubule is the AQP1 type. This channel accounts for high water permeability in the proximal region of the tubule and is always open. As for the distal tubule, AQP2 channels are present to account for the variable water reabsorption. This channel is regulated by a hormone called vasopressin. The reabsorption of water in the proximal tubule is driven by the compartment of hypertonicity in the lateral spaces. This hypertonicity is accomplished by the basolateral pump, which works to actively extrude sodium. This causes sodium to accumulate in the localized region within the lateral space, while also quickly diminishing in both the tubular cells and tubular fluid. The result of the pumping activity creates a concentration gradient, which then allows the passive flow of water from the lumen into the lateral spaces. As fluid accumulates in the lateral spaces, it creates a buildup of hydrostatic pressure, which in turn causes water to flush out of the lateral spaces into the interstitial fluid, where it can then finally enter the peritubular capillaries. Aside from sodium, water can also osmotically follow glucose. However, the direct influence of sodium reabsorption has a greater impact, so it is more important. The return of filtered water into the bloodstream is enhanced due to the plasma osmotic pressure being greater in the peritubular capillaries than elsewhere. This force, together with hydrostatic pressure, works simultaneously to drive water toward the capillaries. By such means, 65% of filtered water is passively reabsorbed once it reaches the end of the proximal tubule. Water movement can only occur if the membrane is permeable to water. I have included an image of a nephron structure on this slide to hopefully provide a visual understanding of the different parts that are permeable and slightly impermeable to water. The proximal tubule is where water permeability is always high. It is through this portion that water is reabsorbed rapidly as the solutes. Conversely, the loop of Henle is where water permeability is always low. This means that water is most likely not reabsorbed in this particular region despite a large osmotic gradient. As for the distal tubule and collecting duct, water permeability can be high or low depending on the absence or presence of the antidiuretic hormone. Like water, urea reabsorption occurs passively and is linked to active sodium reabsorption. Urea is a term used to describe any waste product that come from the breakdown of proteins. Urea reabsorption is enhanced by a concentration gradient formed by the osmotically induced reabsorption of water in the proximal tubule following active sodium reabsorption. When water is extensively reabsorbed in the proximal tubule, the original 125 milliliters per minute of filtered water slowly reduces until only about 44 milliliters per minute of fluid is present in the lumen by the end of the proximal tubule. This causes some substances to not be reabsorbed, so they progressively becomes concentrated in the tubular fluid. Among those substances is urea. The concentration at which urea is filtered at the glomerulus equilibrates the concentration in the plasma by which it enters the peritubular capillaries. About 125 milliliters of filtered urea is present at the beginning of the proximal tubule. This amount is multiplied 3 times towards the end of the proximal tubule, which means that the urea concentration in the tubular fluid now surpasses the urea concentration of the capillaries. As a result, a concentration gradient is established where urea can passively diffuse into the peritubular capillary plasma from the tubular lumen. The walls of the proximal tubules are slightly permeable to urea. For such reason, only about 50% of filtered urea is passively reabsorbed. This means that with each pass through the nephrons, only half of the filtered urea is actually eliminated from the plasma. This removal rate is more than enough. Urea is not as permeable in the tubule as water, so sometimes its reabsorption is facilitated by specific urea transporters. Some of the conditions affiliated with urea reabsorption and filtration include and are not limited to impaired kidney function and severe renal failure. With impaired kidney function, only less of the half of filtered urea is removed. Severe renal failure, on the other hand, was studied and found to be associated with elevated urea levels in the plasma. Although the most detrimental effect and consequences of renal failure is not attributable to the retention of urea in the blood, but rather to the accumulation of hydrogen and potassium ions, health professionals and researchers still refer to renal failure as uremia. Aside from urea, there are several other filtered waste products that are concentrated in the tubular fluid. These waste products cannot be reabsorbed passively, meaning they cannot just leave the lumen and move along the concentration gradient. That is due to the fact that they cannot diffuse through the tubular wall. Urea molecules are the only waste products that follows passive reabsorption because they are very small in size. The other waste products, such as creatinine, uric acid, and phenols remain in the tubules until they are excreted in highly concentrated form in the urine. This process of excretion is able to proceed at a satisfactory rate when renal function is normal. That concludes my presentation for today. This last slide provides references to the sources I used throughout my presentation. I hope this PowerPoint video helped provide a better understanding of the different mechanisms of recovery, as well as the processes of reabsorbing water and urea. Thank you so much for your time. God bless.